before we start the big long paper, you're going to be writing on a separate sheet of paper, just your normal notes paper, because we have to review chromosomes and genes really quickly. That's that key vocab, and then we'll talk about meiosis as a whole with all the pictures you're going to be including. So don't write on your big long paper yet. Write on some normal paper first. But I have a video for you first. Okay, the video I really like for genetics. It's kind of cool. But these are just FYI, quick reviews. Okay, organisms have thousands of genes. Okay. All these genes code for your eye color, your hair color, your height, your weight, your blood type A, B, O, the acidity of your stomach acid, the acidity of your saliva, if you have freckles, if you have a big nose, a small nose. I mean, they code for everything. They code for all the physical traits you have, okay? So that's why we have so many of them. But where do the genes live? They live on chromosomes. You remember this? Yes? From like probably 8th grade? Okay, with me. You're not working on a worksheet right now. Thank you. The word homologous chromosome. What did a homologous chromosome look like or represent? Anybody? What does homo prefix stand for? Same. Okay, what did this mean? Huh? You're halfway right. Okay, they're two of the same chromosome. So here's a chromosome, and here's a chromosome. Let's say it's got like two little genes right here. Well, this one's going to look exactly the same. It's going to have two little genes right there. So these individual lines are chromosomes, but what do they look like when they're replicated? This is the unreplicated version. What do they look like when they're replicated? They have an X, right? They have two sides to it. So these are the unreplicated. These are lovely X's. There's a little, I'm quite sure what that is. Let me go a little better here. There we go. There we go. And there's the two lines, there's two lines, and then there's two lines, there's two lines. So these are still two chromosomes, and this is still two chromosomes. It's just these are unreplicated and these are replicated. They're still called chromosomes though, okay? So they have they're identical, but they don't always have to have the exact same gene. It's just like that gene right there, this gene here and this gene here could still call for your eye color, but one of them's calling for brown eyes and one of them's calling for green eyes. So that's why I say they don't have to have exactly the same exact genes. Okay, so we're going to talk about genetics a little bit more, but we get the light for me, Lauren? This video I love, and it is on Vimeo. It should hopefully play. Let me actually do the one that's already loaded. Okay.
You are part of a diverse...
No. I swear to you people. All that prep work about how much I love this. Okay, I'm going to let it load in the background for some unknown reason. I'll come back in a minute. I've been having some issues. You guys know. You know. Okay, here again are my homologous chromosomes. Yay. Okay, they're chromosomes. All right, you remember the words diploid and haploid?
Yeah.
Okay, the definitions I just want you to rewrite this one part of it, because some of us were so confused when I talked about these earlier. Rewrite this one part. Cell containing two sets of chromosomes. Just that's fine. I think you know diploid are or somatic or body cells are the ones that we call diploid cells. If you don't know that, write it again, but I do want you to write down this. Diploid, cell containing two sets of chromosomes. And then haploid, same part of this definition. Haploid cells are the ones that contain half of it. So instead of two sets, half of two sets would be one set. So a cell containing only one set of chromosomes. And the two different types of cells we talk about with these are like normal body cells, like hair, skin, stomach, bone, toe, fingernail, whatever. Those are the diploid cells. And then the haploid cells are going to be only the reproductive cells, eggs and sperm. That's what we're going to hit a lot today. I'm going to freeze up for a second. See this darn video wants to play. I'm going to let it buffer more. It's only buffered like another 20 seconds. Oh, the struggle. Okay, good with haploid, diploid? Okay, so we're going to talk about sex reproduction, which would we be talking about diploid cells or haploid cells if we're talking about sex? Haploid, thank you. Those are the ones that are the gametes. Gametes mean baby making cells, okay? Sex cells. And you can see this fertilization happening. Little sperm's going, hey, how about we get together? And I will fertilize you, and we'll make a cell, also known as a zygote. Zygote, when these two things, sperm plus egg, makes zygote. That's what all of us were right when the fertilization thing happened. Okay, that's a good word to know. Okay, we already know about mitosis. Don't write that down. We already know about mitosis. What we're going to be talking about is meiosis today. You see, the meiosis is different than mitosis. How do I remember the difference? Mitosis, my toes are growing. My toes are what kind of cells? Body cells. So it's just going to make exactly the same types of cells. It's going to make just some more skin cells to cover my toe, or more toenail cells to cover the toenail, whatever. Okay, so mitosis, my toes are growing. How do I remember meiosis? I go, meiosis, my ovaries. I know some of you guys can't do that, but meiosis, my ovaries. So I'm talking about my eggs. Okay? You guys can be talking about your eggs too, or some other person's eggs. I don't care whose eggs you're talking about. But we're talking about eggs and sperm here. Okay, sex cells. This whole process is going to be making those haploid cells. Okay, so that's why we had to review what haploid and diploid meant, because meiosis makes haploid cells. So that purple definition right there is really important. Yeah. Oh, what is it? Where is it?
Number of
I stutter in my notes sometimes. Okay. Yeah, in my head too. Yeah. Producing cells with half the number of chromosomes. Haploid cells. Okay. And you can see the difference. I'm going to show you that difference. You guys done with that purple definition? Some of us, I'm going to write, just say right there a little bit. So we're only talking about stuff in sex cells now. That's today's lecture. So look at mitosis. You go from how many, you can barely see that. There's 4 chromosomes in there, okay? These are the single ones. Then it replicates, and it goes through, prophase, metaphase, anaphase, telophase, cytokinesis, and it still has 4 chromosomes in each of these. You can't see them very well, but they're there, and it says diploid and diploid, so that kind of makes sense. That's mitosis. That's easy peasy, making new cells to repair old dead ones. Meiosis, my ovaries, goes through 2 cycles, okay, or 2 separations. We start off with 4 chromosomes. They're in that little single phase. You replicate them. They go here. They go through 1 division. They have 2 chromosomes here, but they're still replicated. They've not been, I'm going to put replicated here too. They've not been separated. And then they go through another division, and we end up with 4, what kind of cells? Haploid or diploid? Look at the letters. We have 2 N, and then we go down to 1 N, and so that's 4 haploid cells. So if we start off with 4 chromosomes, over here you can barely see it, but they're ending with 2 chromosomes. So that's why I went from 4 to 2, so it halved. I'm gonna leave that there for a second, but the picture that I'm gonna have you guys illustrate is like 20 cells or something like that. It's kind of large. Yeah. Right there? There? Oh, I'm sorry. What'd you say? That one's not, it doesn't give you much detail at all. Go ahead and jump in with this, okay? So it doesn't give you much detail at all. We're going to talk about the individual. We're going to talk about like prophase 1, metaphase 1, anaphase 1, telophase 1, and so on. But some of the stuff's the same, so I'm not going to go over every detail, because you guys know mitosis like the back of your hand, so we're good. All right. Okay. All right, so do not write this yet. But you'll notice we're going to go through interphase first. I want you to write down someplace 1 interphase. Because you only do interphase 1 time, because if you do interphase twice, you're replicating the DNA twice. And that's dumb. If you're trying to make half the number of chromosomes, it's going to be counterproductive to what you were trying to do and make half the number of chromosomes in your sex cells. So you only do interphase once. You go through prophase, metaphase, anaphase, telophase, stop, and then those 2 cells that are made, this one and this one, 1 and 2, they go through prophase, metaphase, anaphase, telophase again. When that happens, it ends up making 4 cells. So here's what I want you to sketch out. Okay, we're gonna fill these in. Okay, so you want 1, 2, 3, 4 circles, and then a little cleavage furrow thing forming. I'll give you a minute to sketch those. Yes?
Are we going to be writing inside the circle?
Yes, you're going to be writing inside the circles, chromosomes, and a little bit of detail. That's why you have a big paper. This is on your big paper now, okay?
Oh.
Wait, okay.
I didn't get it. Oh.
We're on this week. You don't have a big paper from yesterday? I mean, I do, but I didn't know. So that's what I'm having you do a big paper because you got lots and lots of space for big paper. Your notes is going to be kind of crunched together.
He wants to do it on the bottom side?
Now it's on the back.
Okay.
On the back, so it's separate. Where you guys stick with me?
I was waiting. Some weird else for them.
Look at actions. So go ahead and leave yourself, so you got lots of space, okay? You got lots of space on paper. Space it out some so we can actually write information in the circles and underneath the circles. Jalissa, start for me. Start reading your book right now. Yes. They would be on the back side. Mitosis is on one side, meiosis is on the other side. If you want like a different color to write for your chromosomes or something, just like this. Anybody want a different color? Anybody else? Okay, we're gonna stay here on. Oh. Yeah, I'm getting ready to move it up right now. Okay, so you should have your top 4 with your little weird lumpy thing. And then you draw your arrows too, because these 2 are going to separate into the 2 cells right here. And then, can I go up some more? Can I go up past the green? Yes, we all should be there at this point. Okay, so there's the green over there, and then you break into the 2 cells here, and they go here. I'm getting that pencil pen. Let me see if the video is loaded. Oh, it's starting without us. Alright, I'll give you one more minute to sketch that. And then I'm going to talk about each phase a little bit, and we're going to add images as we go. There you go, take a little seat. That's what, and you've got space to write underneath each one. Beautiful. Beautiful. It's alright, take some time learning to write. Alright, my perfectionist, I'm going to leave this page in a second. I'll be coming back to it every time. But I want to go ahead and talk about the phases. Thumbs up when you get your circles drawn. These circles are plant cells or animal cells? Animal cells, definitely, because they are what shape? Circular. Thank you. Okay, let's go ahead and label. That's what we'll do. Very, very first phase. I'm going to just write letters and not the whole word. First phase is interphase. What does interphase we said it looked like? Blob. Okay, here's that blob. Blob, blob, blob. There we go. Just a bunch of chromatin floating around in there. You know that G1, S, and G2 all take place in interphase. That's when the duplication of the chromosomes happen. Now, next phase is going to be prophase 1. So P1, prophase 1. Alright, what do you think comes next? Nope, not prophase 2. Same same sequence. So, thank you. Metaphase 1, then anaphase, anaphase 1. Okay, so what's this weird thing happening here? Telophase 1. Good. And why is that telophase? How do you know for sure what's starting to happen? It's starting to, yeah, we call that pinching. Cleavage fur. Thank you. So the cleavage fur is starting to happen. That's why we see this pinching action happening here and here. Okay, so then when it finally splits, that was called what? Cytokinesis. I'm just put cyto there. It's not really cytokinesis 1. It's just called cytokinesis. Okay, so this whole prophase 1, metaphase 1, anaphase 1, telophase 1, that whole 4 cells, that's totally called meiosis 1. Okay? We've gone through the first steps of meiosis. Now, each of these cells, this one and this one, they're going to go through, both of them are going to go through meiosis 2. Okay? So I'm going to put a label, I don't know, I'll put a label like right here. Meiosis 2. I don't put M2 because that you might see that as metaphase 2. Okay, so this one and this one, they're both going to go through it. So every time I'm going to label both of the different types of meiosis. So now this cell is my beginning. I'm going to go to prophase 2 on this one and prophase 2 on this one. You got me? So I did prophase 2 on this one and on this one. Because both sets of cells go through those exact same stages in meiosis 2. Then next is metaphase 2. I'm going to put meta so you know I'm not talking about meiosis. Metaphase 2. Thank you. Anaphase 2. Anaphase 2. And then we have the weird blob or weird pinching action going on again. So where are we at? Telophase 2 specifically. That's on this one and on this one. And then last steps. Right, cytokinesis here and cytokinesis here. Feel accomplished, man. We got almost everything drawn set for the chromosomes. Thumbs up when you're all the way through both cytokinesises. Okay, I'm gonna wait just one more second here. Okay. If you're still, got a second here. Notice that I'm gonna put a circle in blue around my final cells. You maybe want to draw a circle or a square around your final cells. So there are 4 new cells produced. The very, 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 very final product. So I'm going to put final. We have 4 new cells produced. All right, how is that right now? How is that different than mitosis? Yeah.
Mitosis only makes 2 cells.
Right, mitosis makes 2 cells. Meiosis makes 4 cells. Okay, move into chromosomes. I'm going a little slow because I know you guys like to draw on your stuff there. All right, let me tell you details of each of these first. So, not each, just some. Prophase 1 is my first one. You might have room to write it on your drawing. You might not. So write down it elsewhere if you need to. Yeah.
After the T1, are both those new cells cytokinesis or just one?
Yeah. Both of these are produced from cytokinesis, and then they separately go through meiosis 2. Okay. Interphase. You already got your blob drawn, right? Yes. Write down DNA copy because I can't let you forget that DNA has to be copied there. You can write it on your drawing or someplace else. It just depends where you have room. Some of us drew huge murals. Some of us drew like itty bitty murals. Okay. After interphase, in prophase 1, the DNA coils. Okay, so in your DNA coiling, that's just going to mean that it's going to shrink down and actually condense in a way that you can see it. But this is very, very important. We're going to have a star. Under prophase 1, right, crossing over occurs. Crossing over occurs. And we're going to draw in our prophase bubble the same stuff we would do in prophase mitosis. This is prophase 1, by the way, so P1. You've got your nuclear membrane. I'm going to draw them as we go, okay? Nuclear membrane, that's disappearing. And then let's draw 4 chromosomes total. So 4 chromosomes. I'm going to draw 2 large ones and then 2 small ones, just so I can distinguish between them. 4 chromosomes total. Notice though, I've got them, those big X's next to each other and the little X's next to each other. What happens is they can actually get super, super close to each other, and they kind of tangle their arms and they cross over. I used to ask people what they thought crossing over meant. They thought the cell died. It's like crossing over to a new world. I don't know why. But now we just see that these little crossing over areas, they actually like switch their arms. So like if I had somebody stand next to me and I was my ex, and they were standing next to me, we had our legs and our arms tangled, like I would get their arm, they would get my arm, I would get their leg, they would get my leg. Okay? So actually what that means is it exchanges DNA. Okay? You'd have to know that happens in prophase 1. That is very important. That's why I'm going to start there. And then, what does the word tetra mean? The prefix tetra. Mono, di, tri, tetra. 4. Okay, tetra means 4. So this is called a tetrad because how many like sides to the chromosomes do you see? 4. You see 1, 2, 3, 4. Good. That's why it's called a tetrad, that little mixing up thing right there. Okay, then, oh, I'm sorry, I didn't draw. Well, I did. I drew prophase 1. There we go. Let's do metaphase 1. Metaphase 1 is really similar, except now your pairs of chromosomes align. Pairs of chromosomes align on the equator. So if this is our cell, the nuclear membrane is gone. Now, instead of lining up, don't draw this yet. This was how it lined up in mitosis. Yeah?
Yeah.
Now, those are single file lines. It says pairs of chromosomes line up together. So now we have tetrad here and tetrad here. They line up together in a line. See how that's different than my mitosis metaphase? It's turning inwards. Okay, so that's really important. What's attached to these? Spindle fibers. Thank you. Okay. So you got your spindle fibers that are drawn in there as well. You with me? Yep. Can I go on? Wait a second. Okay. So they line up in their pairs, and that's really important. That's why I'm going to start by that one too. Write down this information as you're drawing the pictures. Right. Forward. Onward. Yes? Oui? Okay. Here's that lovely, look at those crossing overs. Woo! They're getting all tangled up. So you see your little homologous chromosome pairs. They go, hey, let's switch our arms and legs, and they switch parts of their body. And you can see the green and the yellow get mixed together. This right here is actually really, really good. Wherever you wrote the word crossing over, I want you to say increases genetic diversity. That is the reason that crossing over occurs. So that every baby your mom has does not look exactly the same. Okay? Some parts of the DNA are going to be switched around, so depending on what egg is going to get used, different genes can be present. That's why you might look a little bit like your brother or sister, maybe a little bit, but not like, oh my gosh, we're identical, like we're twins or something. Okay? Obviously, twins can't occur. Totally different story there. Okay, anaphase 1 and telophase 1. Anaphase 1, I need to draw this. Go ahead and write down what's in red right there. I need to give myself some space. Under under the symbols or around the symbols or someplace if you have room. So, anaphase 1, you've got your cell. In anaphase, in mitosis, what happens to the chromosomes? They split, okay? Originally in mitosis, this got split to this and this. That is not happening here. What happens is the spindle fiber pulls this one X this way and this other X this way. And then this little X this way and this little X this way. So what has been separated? The pairs, thank you. The homologous pairs are separated. not the sister chromatids. The sister chromatids were separated. Probably have some gene mutations up in those eggs and sperm. Okay, that would be not great. Okay, so the sister chromatids stay attached. That's why I have this in red. And the homologous pairs are separated. Super important. That's why this one's so different. Okay, telophase, what do you think happens? What starts to happen in the middle? It starts to do what? What's this thing called?
The cleavage furrow.
Cleavage furrow forms. You get your big X over here and your little X over here, your big X over here and your little X over here. So it's going to start to suck, it's going to start to pinch in the middle. Can't really erase that very well. You guys already have yours drawn. There we go. Alright, so then what would happen in cytokinesis?
They'd be fully, yeah.
Thank you, fully separated. And then write a note underneath cytokinesis where we wrote, we had those 2 cells drawn, you got your chromosomes. Notice, are these chromosomes single or duplicated still? They're still duplicates. So if it's still duplicated, that's really, really important to write under cytokinesis. Okay. Telophase. Sorry, you already got that. My bad. Let's do meiosis 2, but I'm not going to give you notes on all these because this is super easy. Meiosis 2 looks just like mitosis. Okay? You're just going to continue on both tracks. So both of these cells that we made here and here, they both do exactly the same thing. That's why we have to draw both of them lined up there. So I'm going to show you guys how to draw these here. Here we go. Let me go back to my actual drawing. Was it here? Okay. So what's right here in these cells?
Just the
Big X, little x, big X, little x. All right. This big X and little x is going to be here. I'm just going to try and draw them in 2 different areas because I don't want it to look like I'm lining them up yet. So whatever I draw in P2, so prophase 2 here, I'm drawing exactly the same thing for prophase 2 here. Yes? Because they're both identical, or showing the same things here. Metaphase, what happens like normally in mitosis?
they're lined up.
In the middle, in a single file line, right? So single file line here, single file line here. All right, what happened in anaphase now? We had our stuff in the middle.
Split.
What is split? Use your words.
Wait, in mitosis?
Well, if this metaphase 2 of meiosis 2 looks just like mitosis.
So then there's.
What is split? Say the 2 sister chromatids. Thank you. The sister chromatids are split. Sister chromatids are split. Oh, and I totally forgot to draw what's supposed to be attached to these babies. Spindle fibers here. Oops. And here. And here. And here. Okay, there we go. Alright, we're almost done. We've almost made eggs and sperm. Okay, what's next? Telophase 2, yeah? Okay, now the cleavage fur is starting to form. I'm just going to draw these kind of like haphazardly in here. They're just chilling. Big one, little one. Oops, there we go. And then we got over here. Big one, little one, big one, little one. What's starting to reform around the chromosomes though? What's it called? The nuclear membrane. I'm just going to put it as dots. So we got nuclear membrane, uno, dos, tres, cuatro. Okay. Now the real fun begins. And what have we got left here? What's left?
The 2 cells.
There's the 2 cells from this one and the 2 cells from this one. And the nuclear membrane is totally back. I'm going to draw my nuclear membrane about both of these, or all 4 of these chromosomes, or those things, cells. Okay. They look the same, but they're not. What happened in prophase 1? I just didn't show it on my pictures. Prophase 1, you write it right under prophase 1. Yes, the DNA exchange called Crossing over, okay? Called crossing over. So technically what this could look like is something like this. And then like this, little green spots, little black spot. Okay, it could look different. So if I draw it like that, you see that these 4 haploid cells, they are diverse, yes? So new cells produced, write down the word haploid and the word diverse. And then I'm going to show you that video because it's already loaded. Diverse. Actually, take a break after you get this done. Take a quick, like, 1 minute break. Walk around the room. Stretch, yoga, wusa, whatever. I see a couple of those that need to get up. Go splash water in your face. It's Friday. Do a cartwheel. Upright leg. I want to see your beautiful drawings. Oh my gosh, yours is Picasso's. Lovely. Are you guys like holding hands for a reason? Oh, how cute! Holding hands over there. Oh. Hug it out. Hug it out. These are awesome, guys. Good job. Okay, cross your fingers. The video will work, people. Okay. Oh, what is this mess? You've only loaded the 2 minutes?
I think we can play it with her.
Okay, here we go. Ready? Lights. Camera action. Sorry, Miss Jackson. Okay, I'm not gonna mess with it. Leave it like that.
You are part of a diverse.
No! You.
We just pause it for a minute.
It's been paused for like 20 minutes.
Wait, no, wait.
Okay.
Yeah, turn HD on.
You guys are super smart. All right, let's try this again.
Yeah,
Let's see if I refresh, it has to rebuffer, and I'm not cool with that.
we just started the rebuffer.
I know. It's got issues. I could probably find this on YouTube. Vimeo is causing me some probs. Okay, not touching it. Can we get past the first 3 seconds?
You are part of a diverse array of organisms that inhabit the biosphere on Earth. Bacteria, plants, animals, and you are all carbon and water-based cellular forms of
Good part of it.
For my phone?
Yeah.
Or 18 things you should know about genetics. Got it. 18 things you should know about genetics. YouTube? Aha! Vimeo. We'll talk later. Let's see if this is the same video. This looks... Okay, we're just gonna keep going with this. We've seen this part like 5 times now.
You are part of a diverse array of organisms that inhabit the biosphere on complex organization and heritable bacteria, plants, animals, and water-based cellular forms with complex organization and heritable genetic information. So, are you curious what makes you, you?
Yes!
Your genes make you, you. Genes make up the set of instructions that are given to you from your mother and your father. These instructions are in almost every cell of every living thing and are stored on extremely long strands of chemicals called DNA. DNA stands for deoxyribonucleic acid, and these long strands are called chromosomes. Chromosomes are made up of many genes. One gene is a specific sequence of DNA on a chromosome that provides particular cellular instruction. DNA is shaped like a ladder that's been twisted, which is called a double helix. The steps of the ladder are made of 4 main chemicals called bases: adenine, thymine, guanine, and cytosine. The particular order of A's, T's, G's, and C's is extremely important as it determines whether you are a banana, rhinoceros, or a human. It also determines whether you have brown hair, blue eyes, or green skin. The sides of the ladder are made of alternating sugar and phosphate molecules. The entire DNA sequence is called a genome, and in humans it's 3.2 billion base pairs. This would take 9.5 years to read out loud without stopping. It would take someone typing at 10 words per minute for 8 hours a day, 50 years to type the entire human genome. If you unwrap your DNA from one cell, it would extend more than 6 feet long. If you unwrap all of the DNA in all of your cells, it would reach the moon 6,000 times. All 3 billion bases of your genome would take 3 gigabytes of computer storage space. Your entire DNA sequence would fill 200 New York City telephone directories. You are 99.9% identical to the person next to you. Only 0.1% of our genetic makeup differs. Less than 2% of your DNA carries recognizable instructions to make proteins. The rest is called junk DNA, as it does not currently appear to code for anything. It takes around 8 hours for one of your cells to completely copy its DNA sequence. You are you because of your genes. Now you know more about this microscopic entity that makes us all the same, but grants you with distinct differences that make you unique.
That's how I'm gonna show it. Like, it was good. Yes.
Why was there countdown in the left bottom corner?
18 things. I think it was like doing 18 things. Yeah. Okay, oop, you don't want that. Let me see. Let me give you guys a break for a second. I have pictures and pictures and pictures. Do I want to do these pictures? Yes, please. I've got 1, 2, 3 more slides. You want to do slides right now? Can we do it real fast?
Yeah.
Let's get this done. Okay, I'll show you the pictures momentarily. All right, the whole purpose of meiosis is not to make egg and, well, okay, it is to make egg and sperm. Write it someplace other than your pictures, yeah. Write this on, like, normal paper, okay? Normal paper, not that abnormal paper. Okay, you gotta know, 1, the purpose of meiosis is to make egg and sperm, and 2, well, not all the eggs can be exactly the same, and not all the sperm can be exactly the same. So, this whole mess creates a ton of genetic diversity. This is very, very important to know or relate to for meiosis. This is different than mitosis, because mitosis makes these things called identical cells. Because you don't want your toe cell to look different than the other toe cell, because that would be weird going to swim school, right?
Yep.
You don't want to tie up hair or make you. You want to make sure that your cells and mitosis make identical stuff because you need to rebuild those exactly the same. But meiosis, you want diversity. It's actually really good to have diversity. So the 2 phases that caused diversity to occur were, refresh your memory, when did crossing over occur? Prophase 1. That one is going to definitely help create genetic diversity. That's mainly the one that we're going to be talking about. There's another one that's anaphase because it can separate these things into different cells. Yes, ma'am.
I know some with 2 different colored lines.
That is called hyperchromism or heterochromism. It's actually a genetic disease.
Oh, well.
Yeah.
Actually, they weren't the ones brown. Yeah, I don't know, one of them was green, one of them was brown.
Is no contacts?
No contact. And, like, I guess somebody's literally stuck a fork in the outlet.
Oh.
Oh, so he electrocuted himself.
Yeah.
Burn the pigment or something? Maybe it's like.
It changed the color.
It can, okay, what can happen is there's environmental factors that could change the expression of your genes. So maybe that caused his genes to go, wow, we need to make some brown eye color in this one, but not this one. It could have been environmental. Or it could have just developed over time, because most babies are born with blue eyes. Most babies. Your eye color changes a little bit after. FYI. Okay. So, this other part here, genetic variation is advantageous. What's the word advantageous mean, Sam? It's not.
Advantage.
So what is an advantage? What do you mean? Like, if I have an advantage over you, what does that mean?
There's something better than me.
Yeah. So, like, why would this be advantageous? Because sometimes when you have diverse genes, maybe one person inherits genes that means they're really athletic, so they have a really great advantage to play sports, where somebody else inherits genes that makes them really good at math, helps them, like, do mental calculations better. They have an advantage in the math world. Or it could be a giraffe that inherits a gene to make their necks longer. So they have all the food they want at the tallest trees where everybody else has got to like stretch to get the lowest part of the limbs. So things that are advantageous help you survive in your environment. So we can talk in the human world, but most of the time we have like technology and stuff that gives us advantages. In the animal world, it's more about what they can do and how they can survive. Good? See? No, wait, one more second there. It's also known as no. Okay. You want to talk about anaphasia now? Okay. I'm just going to mention this, and I want you to write down these two words. Write down the word non-disjunction. Non-disjunction. That is a big word, right? And tell me that non-disjunction can happen during anaphase 1. So the two blue words right there. And so non-disjunction can happen during anaphase 1, and that can cause Down syndrome. That's just one example. So that's one that we all know what Down syndrome is, yes? Okay. So what happens is whenever the chromosomes are lined up in metaphase, like here, we had our little pictures, and we said, okay, normally, they're in their tetrads, they're in their fours, they're in the pairs. When we said, okay, anaphase happens next, we said, okay, this one goes this way, this one goes this way, this one goes this way, this one goes this way. But what? Yes.
What's the definition of syndrome?
Syndrome, condition. Yeah. That's kind of like an interchangeable word. Okay, so what can happen is, yeah, we can have this chromosome go this way, this one go this way, this one go this way, this one go this way, then it splits. Okay, even numbers of chromosomes were good. But what if accidentally the spindle fiber, instead of pulling that one to the left, it leaves it alone and it leaves it with this one? Yeah, because how many chromosomes are in this cell then? 1. How many chromosomes are in this one? 3. So then this person's going to inherit an extra chromosome whenever they get that cell later on, and now one's going to inherit one less. And so if you don't have enough chromosomes, or if you have too many chromosomes, you generally will get lots of different types of diseases. Down syndrome is one of them. Fragile X syndrome is another one. Maple urine disease is another one. Yemenite deafblind hypopigmentation syndrome, leukemia inhibitory factor. You can get all kinds of. That whole thing is full of diseases. Those little words are just diseases and disorders.
Wow.
Yeah.
What's maple urine syndrome?
Maple Europe. Your urine smells like maple syrup. It actually means that you're excreting a lot of sugars, and it's really bad because your cells aren't getting the sugars. Yes, sir.
Do you know if bipolar disorder can be led back to X or
Like a maternal.
yeah?
I don't know for sure if that's maternally inherited. I don't know if it's which chromosome it's on. I can look on that poster. It's going to take me a day or two because it's really long. I can look that up though. Okay, but sometimes that is genetic. At least they think it is. Okay, so this can cause something called trisomy. Well, if I can go down here. All right, so this, in the end, you get 1, 1, 1, 1. That's normal, but if you don't separate your chromosomes right, you can get 1, 1, 2 chromosomes there, and then no chromosomes there. That can cause problems. That's what non-disjunction is. And that can actually cause quite a few problems. We know this XX female, XY guy. I don't really, I don't know if it's something you're aware of, but XY guy
Attention teachers, all 10th graders
is
are
something that you
returning
need to know.
back to class at this
We're
time.
going to take those
Thank
photos
you.
off on the YouTube, yeah.
Yeah,
Why don't you see it? You actually do see it. Whenever it's these right here, this is one chromosome and this is another chromosome. The reason that we draw chromosomes like an X, like that, it's not because it's the X chromosome. It's not the X chromosome. It's just 2 chromosomes that are linear put together. This right here is how you represent every chromosome in the body when it's replicated. What the X chromosome actually looks like is like this. That's the X chromosome, and that's the Y chromosome. They look exactly the same as all the other ones. It's just the Y chromosome carries like half the genes, So you guys don't have as many genes as we do. That's why a lot of times people say females are superior to men.
Go ahead, Brian.
That's what people say. I'm just saying. We have more DNA. She's coming back from planning and all in the room. Thank you. Bye. Okay. Woo. All right. So you know XX girl, XY guy, yeah? Okay. My people who have been here, I'm going to let you guys go ahead and work on two things. We have some stations set up, and I'll explain that in a second, but you can work in groups at the lab stations. Wait for it. You're going to have this cell cycle worksheet, which is not too hard. On the back side, you have a Venn diagram and a compare-contrast of the daughter cells, etc. Question? Yes, this is yours. So you guys can work together, and you can finish your cell plate if you've not finished your cell plate. I have the brass brats right behind Reagan, okay? So the last, 25 minutes of the class is yours. Please get this done. If you've not grabbed a worksheet, grab a worksheet. And if you have to finish your cell plate, go to the back. People up here who are just coming back, you have a YouTube video to watch because I'm not re-lecturing. Let me upload it real fast.